collaboration. So many people from many states are coming to see you. So like if we have a very important person talking to us like you, we take the opportunity to join all the PhD programs in Brazil, right, Tetsu?
Yes, yes, Brian. Thank you for your presence here in our event. It's a great pleasure to meet with you. We are very excited about your seminar today, your lecture today.
And do you remember Rafael? Yes, Brian.
No. No.
Very well, of course.
Very well. So how long have you been there, Rafael? Some 15 years ago.
Yeah, well, I arrived there almost 10 years ago, but I was there for my whole PhD, so almost 5 years.
5 years. Okay. So what's your expectation, Rafaela, for the Brazilian bioinformaticians community?
Okay, so I guess I would like to also thank Brian for being here. It's really a pleasure to have you here, Brian. And for people in the molecular modeling community, I think Brian really doesn't need much of an introduction. So I'm gonna be brief here and just say a few things. So Brian has been a faculty at UCSF, the University of California, San Francisco, for about 20 years. He was also a PhD student and a postdoc there. And in his PhD, Brian worked with Tak Kuntz in the development of DOC. So those were really the early stages of the program DOC. And then afterwards, he moved on to a experimental postdoc with Brian Matthews. And that's very interesting because we can see from his PhD and postdoc how Brian got expertise in these two areas, which they are very present in his lab nowadays. So basically, Brian continues to develop DOC. Now we are in version 3.8, which is very nice for virtual screening, has some very interesting features. And Brian and his group have contributed a lot in the field of docking, virtual screening, and structure-based drug design. In terms of virtual screening, we have lots of successful applications of this technique, and also a work to aid the community in terms of setting up database of commercially available compounds, also of decoys, which are very important for careful and rigorous virtual screening. So Brian has worked with model systems that help to aid in the development of scoring functions and improving DOC. Also, there is a lot of work with MPC beta-lactamase. And more recently, for more than a decade, I guess, he has worked a lot on GPCRs. So in the past 5 years, or approximately that time, Brian has reported lots of studies with ultra-large virtual screening, showing that it has been successfully applied to discover new leads for several targets, and also with a very critical analysis of why should we care to work with these large databases. So Brian has lots of publications in very high-profile journals, several awards, honors, and it's really a great privilege to have you here, Brian. So thank you very much, and I'll turn the word to him.
Well, it's great to be here. Thank you for those kind words, too kind. I hope you can see my screen.
Yes.
This is as simple as it's going to be right now. So I just thought anyway, I'm thrilled to be here. So wonderful to see Rafaela, who was a tremendous force in our lab, did seminal work on understanding what large library docking could achieve, how it compared to high throughput screening, and all in the very good cause of inhibiting cruzane. We were very sad when she left, but very happy for her. Here's what I'm going to talk to you about today. I'm going to introduce this idea that Rafaela referred to, ultra-large library docking, and how it works a little bit. I'll tell you about how one story, we'll go into one story in a little bit more depth, how screening a large library computationally, structure-based screening can lead to better pharmacology. We'll talk about the calcium sensing receptor. I'll talk about some of our problems. One in particular, we struggle with improving compounds, and we've taken a step back to understand, why we're struggling and what underlies the ability to improve a compound for medicinal chemistry. And then finally, at the end, I'll return to this idea of, are we actually improving things with the large libraries? Or maybe, we're now docking 6 billion molecules. Maybe we can stop. Maybe we don't have to make them any bigger because 6 billion is a lot. But what I'll try and convince you of is that, in fact, we should continue to grow our libraries, everybody's libraries, because as they grow, we get better and better results. These are my conflicts. Okay, I'll start with an introduction of large library docking, really as it was 5 years ago. So what we do is we start with a library of small molecules, up to 5 years ago, that was about 3 million molecules. And it was important, still is important, that they be readily available, because we take each one and fly it into the binding site and score it for how well it fits in the site. But in calculating docking scores or energies, we must make many approximations. And so we get a lot of things wrong. And so our confidence in any particular prediction is low. And so and we expect to fail many times. And to make failure cheap, we insist that we just be able to purchase the compounds quickly. Anyways, each molecule is flown into the binding site in multiple orientations and conformations. About 1,000 or several thousand orientations, rigid body fits of the molecule are calculated. And in each fit of the molecule, multiple, maybe several hundred conformations of the small molecule are sampled. And overall, we might sample about 1 million configurations of the small molecule in the binding site. And we do that for each small molecule in the library. They're scored using physical chemistry-based scoring, for instance, van der Waals interactions and electrostatic interactions. controlled for the solubility of the compounds, the desolvation energy of bringing the molecule out of solution. And at the end of the calculation, we have a ranked list from best fitting to worst fitting of all the molecules in the library. And the big change in the last 5 years has been the libraries have grown enormously. So now we're doing this not with 3 million molecules, but with 6 billion molecules. And a major theme of this talk is the impact of that library growth on the success rates of docking. Okay, how do we get to 60 billion molecules? Well, 6 billion. I said we dock 6 billion. That's true. The size of the library is actually 60 billion. And the reason why we're only docking 6 billion is that the library is expanding faster than we can create new structural models of the small molecules. Here's how it gets to that size. I should say that the idea of virtual libraries has been around for a long time, but we never believed in it. So we would never dock virtual libraries until a few years ago. And what changed our minds is these scientists at Inamin in Ukraine convinced us that they had a virtual library that they could actually make and get to us in relatively quickly and relatively inexpensively. And the way they were able to create this library is that over about 30 years, they had developed building blocks on site that they had in multi-gram quantities. And these building blocks were interesting. They were stereochemically complex. They had interesting fused rings and, bicyclo rings and this sort of interesting looking molecules because they were chemists and they were driven by chemical complexity. And what they would do is they could take pairs or triplets of these interesting building blocks and put them together in, by now, around 200 different reactions. And that in sort of 2-component or 3-component single purification step reactions, that led to this enormous diversity of molecules now into the many billions. So that's the origin of the virtual library. None of these molecules, well, very few of these molecules have been pre-synthesized. They all exist as potential molecules. And our goal, and anybody running a virtual screen on this library, which is publicly available, is to pick out ones from this enormous space that then Inamin would synthesize for the first time. Okay, so what I told you is that docking makes many errors because our scoring functions are approximate and we undersample states. So you could wonder why it should ever work. And particularly, you could wonder if expanding the library could make everything worse, because if the number of false positives grows faster than the number of true positives, if it outstrips the ability of the scoring function to distinguish likely from unlikely molecules, which was very possible, then going to 1 billion molecules could easily make everything worse. So we wanted to test this idea at scale on the sigma-2 receptor. Sigma-2 receptor is sort of what we call a dark horse of pharmacology. It's a receptor that's been long studied, but its role even today is not well understood. And it's even its molecular identity was controversial for a long time. But in 2017, Saf Alon here, who's now at Yale University, working with Andy Cruz, cloned it for the first time. So there still are some receptors that aren't cloned. And then a few years later, determined its structure by protein crystallography. And working with Andy and Asaf, J.K. Liu in our lab decided to run a screen on it for novel molecules. And at the time, the library was 500 million in size. And we did what we always do is we dock each one in turn into the binding site and score for fit. But here in this campaign, because we were interested in testing the method, we did something a little unusual. Rather than only testing, 30 or 40 or 50 high-ranking molecules, we decided to test, actually, we wanted to test 600. So we ordered 600 molecules, each one dissimilar from the other. So a very diverse set, and all dissimilar from known sigma-2 binders. And rather than just picking the high-scoring ones, we picked high-scoring ones and mediocre scoring ones, and even poor scoring ones, hundreds of them, so we could test how well the scoring function could distinguish really good scoring molecules, really good binding molecules from molecules that bind weakly or don't bind at all. So we wanted to test the ability, how a docking score correlated really with whether a molecule would bind or not. That was the real test. And to do that, we had to test, to power it statistically, we had to test many hundreds of molecules. So this is the initial results. Of the 484 that enamine was able to synthesize, we ordered 600 and from this diverse virtual space, and they were able to synthesize 484, which gives you a sense of their ability to accurately to actually make the molecules they claim they can make. Of the 484, 128 were active with IC50s of 1 micromolar or better. And these assays were done by radioligand displacement by Asaf in Andy's lab. So Asaf was able to determine the X-ray crystal structures of 2 of them, and which allowed us to see if the docking predictions were right for the right reasons. Did they bind the way we predicted they would? So here's one molecule. It's basically a 4 nanomolar ligand for the sigma-2 receptor. This is directly out of docking. And in yellow is the docking predicted pose, and in the green and blue is the X-ray crystallographic pose. So this one was as right as we could hope it would be. And Asaf also determined the structure of a second molecule bound to the sigma-2. Here it is, also a 4 nanomolar molecule out of docking. One thing I'll say is you can see that the 2 molecules do not resemble each other topologically. They're pretty different chemotypes. And that's something that we insist on in the docking. We don't test analogs from a docking screen. We're looking for diverse molecules. So this is an example of that. And you can see that here too, the yellow structure is the docking prediction. The blue is the crystallographic geometry. And it's not perfect. There are some differences, but basically right for the right reasons. All the key interactions are made, and it's pretty much in the right spot. So both of these are consistent with the docking predictions predicting the experimental results. Here's what I promised I would show you. This is how docking score correlates with hit rate. And hit rate here means the number of molecules that are active experimentally divided by the number tested. So, up here at around 50%, That means if we test 100 molecules experimentally, 50 would be active, and active means 1 micromolar or better. Many were better. So you can see, and the docking score, the more negative it is, the better. So you can see that the hit rate at very good docking scores starts off at around 50%, even a little bit higher. And then as the docking scores get worse, the hit rates steadily and monotonically decline until they drop to 0, and they stay at, and then they, as the scores get worse, they stay at 0. And which these numbers over the top are the number of molecules that we experimentally tested. So we think that these data and this, the functional form to fit them is well supported by the data. Some of you will notice that this, I don't know if you can see my mouse, I hope so, that this point at the best docking scores is actually substantially worse than the rest of the curve. At the very top, the very best scoring molecules, the hit rate suddenly drops dramatically. And we didn't fully understand this at the time, but what we've come to understand is that these molecules are molecules that cheat our scoring function. This is what we were worried at the beginning, was that because of the inaccuracies in docking, you'd get, as the libraries grow, you'd get more and more of these cheating molecules, and they would mess everything up. And in fact, that's true. But there's very few of them, actually. There's still a problem because they're sort of a fixed percent, a very tiny percentage of the library, but as the library gets bigger and bigger, their absolute numbers grow. This is some, as I'll show you, this is something that we can deal with. But it's for this reason. Oh, the second thing that I'll say is that you notice that the curve, it's a beautiful curve. It looks like a concentration response curve with which pharmacologists are very accustomed. But unlike a concentration response curve, this curve shouldn't plateau. It should, as the scores get better, it should keep improving. But it doesn't keep improving, it plateaus. And so what this teaches is that our ability to distinguish molecules as better or worse among the top scoring molecules stops improving. So we stop being able to distinguish. And that is a problem. And so these are two things I'll come back to: the occurrence of these cheating molecules, these artifacts, and the fact that the curve plateaus. Okay, what I can say is that anecdotally, at least, as the libraries have grown bigger and bigger, our results have improved. And I'll show you that in just a bunch of different campaigns. So an early example of the large library docking was a campaign of, we were docking, this is Reed Stein's work. We docked at the time about 180 million molecules against the melatonin receptor. The best molecules coming from docking were in the low nanomolar range, and we were able to, you'll see with a lot of work, progress these molecules into efficacy in animals at shifting the circadian clock. And sigma 2, I showed you those results. There we eventually got molecules with 4 nanomolar potency. The ones I showed you weren't the ones that we advanced into animals, because we had to fix some PK problems, but eventually we got these, and these molecules were active as analgesics, working with Alan Basbaum's lab. Same thing with the alpha-2A adrenergic receptor. Here, the best scoring molecules were, the most effective molecules had about 10 nanomolar potency directly out of docking. And these were very potent analgesics, non-sedating analgesics, so treated pain. And I should say so here what I'm showing you in Sigma 2, as I showed you before, is the correspondence of the docking prediction with the X-ray result here. There's also two molecules shown. Maybe you can't see it, but once again, the docking prediction superposed on a cryo-EM result. This is work with Brian Roth's lab, where eventually we got mid-nanomolar compounds against the serotonin 2A receptor and showed that those were effective against depression. And another campaign against abuse, targeting the serotonin transporter. And I should say, basically, in all these campaigns, the predicted structure is eventually compared to crystallographic in the case of macrodomain from COVID-19, but usually a cryo-EM result to demonstrate that we're right for the right reasons. And I'm going to go into more detail on the calcium sensing receptor. So the reason I'm showing you all these is that the potencies we achieved here were unprecedented. These molecules were far more potent than we were used to from docking smaller libraries, and often had better pharmacological properties. So those all suggested that we were getting, that the large libraries in which we'd invested a huge amount of work were actually helping us. And I should say, all these projects are huge collaborations with biologists and molecular pharmacologists. Brian Roth, Alan Basbaum. Brian Roth's my longtime collaborator, sort of one of the world leaders, maybe the world leader in GPCRs, along with Kabilka and a few others. Alan Basbaum, brilliant animal pharmacologist. Andy Cruz, I've shown you before. Gary Rudnick at Yale. Peter Gemeiner at Erlangen. My good friend Yorgos Skiniotis at Stanford. Bill Wetzel at Duke. And my close friends and collaborators at UCSF, Ashish Manglik and Jamie Frazier. Okay, so I want to, in the next part of the talk, I want to talk about, I want to go a little deeper into one story, the calcium sensing receptor, and what I mean by better pharmacology. So the calcium sensor. Now, all you bioinformaticians, we pause for a little actual pharmacology. So the calcium sensing receptor is it's a family C, and I'll show you what that means in a second. G protein coupled receptor. GPCRs are called GPCRs because once they get activated, they couple to G proteins inside the cell, and that leads to a signal transduction cascade. The calcium sensing receptor is activated by calcium. As calcium levels rise, this receptor gets activated. And that's important because calcium levels are strictly controlled in the body. They're in healthy humans, they range between 2.2 and 2.6 millimolar and rarely fall outside of that range. And that's because of the calcium sensing receptor's role. If they do rise, the calcium sensor is activated, and in activation, it stops the secretion, it represses the secretion of parathyroid hormone. What that does is it stops reabsorption from bone of serum of calcium, which would normally go into the serum. So activating CASR stops this process, and so you get less serum calcium going into the blood. And it also inhibits the excretion through the kidneys of calcium. So activating CASR reduces secretion of calcium, and so more of it stays it increases the excretion of calcium because it stops parathyroid hormone. So it stops something that stops, and it reduces its reabsorption. So both things lead to lowering serum calcium if it rises. In people on dialysis and with certain thyroid cancers have problems with serum calcium control and suffer from either from hypercalcinemia, which can be deadly. The reason why calcium level is so tightly controlled is because of calcium's role in voltage stability of cells and heart rate. And so if calcium gets out of whack, people die. So to control, and there's a lot of people on kidney dialysis. So to control calcium levels, pharma has introduced drugs like cinacalcet, which is a blockbuster drug, sells at its peak about $2 billion worth of sales, treated a lot of patients, still treats a lot of patients. And evocalcet's a drug that has very interesting properties, but is right now restricted to Japan. They work, both molecules, both drugs act as positive allosteric modulators. This is the structure of the calcium sensing receptor here. It's an obligate dimer, so there's two monomers. The drugs bind in the 7 transmembrane domains of the monomers, and they're considered positive allosteric modulators because the actual ligand calcium binds up here. So they bind at a different site and potentiate the activity of calcium. And basically, so they super activate the receptor. I said there's 2 monomers. Each monomer is different in the activated state, and the drugs bind differently to each monomer. So to monomer A, cinacalcet and evocalcet, though I'm not showing that, bind in this extended conformation, and in monomer B, they bind in this folded over conformation. And only one of these is activate. Only one of these couples to G protein, and it's this B monomer. I'll come back to that. So the problem with these drugs is that they lead to they're sort of too successful. They lead to hypocalcinemia, so too little calcium in the blood, and that also has cardiac effects. And so this is a dose-limiting toxicity of these drugs. And so what we thought was, well, maybe we can find new molecules that bind to the calcium sensing receptor and are effective at potentiating it without these hypocalcemia effects. So what we did, what Fengyu Liu did, a brilliant postdoc in the lab, was screened both a small library. This is like our early in-stock library, the libraries we used to use, 3 million molecule library, and then a much bigger 1.2 billion molecule library. For now, I'm only going to focus on the results from the 1.2 billion molecule screen. So each molecule was docked into the allosteric site and scored and ranked like we always do. And we prioritized about 100 for testing, 74 were successfully synthesized. And of those 74, 24 were active, the best of which was, in the 300 nanomolar range as positive allosteric modulators of calcium sensing receptor. I should say this work just came out last week. The molecules were diverse. We insist that they be diverse. Not all high-scoring molecules are diverse, but we only pick ones that are chemically different from each other, and none of them were analogs of known calcium sensing receptor drugs. But physically, they did resemble CASR. So here's the cinacalcet. So this is cinacalcet, the blockbuster drug. You can see it's got this, naphthalene ring on one side, a phenyl with a trifluoromethyl group on the other side, and a cation in the middle. And our molecules, although they're topologically different, have similar features. So, you know, a benzothiophene instead of a naphthalene, this 2-ring system instead of the trifluoromethylphenyl, and that sort of. So physically, you because they're binding in the same pockets, right? So and interacting with the same residues, they're just doing it with different chemotypes. So what we were left with at the end of the screen were molecules that, you know, at best bound in the, you know, high nanomolar range. So not potent enough to be drugs, and also maybe a little bit greasy. And so what we wanted to do was optimize them. And so just 3 examples. This 425 nanomolar positive allosteric modulator was optimized by about 100-fold. This molecule, which started off at even lower potency, was also optimized by about 100-fold. And this molecule, same sort of thing, optimized by, well, actually, this guy was optimized by 1,000-fold by making analogs. And this was sort of a long process, and I'll come back to that. And the other thing I'll say is that cinacalcet is a pretty high C-log-P, it's a very greasy molecule. That leads to problems in drug discovery. So we were conscious of trying to keep the C-log-Ps to lower levels, to better levels. And I think we largely succeeded. These are the concentration response curves. So now with, I'll just say that it's this molecule, this 30 nanomolar molecule, that we took into animals. With these, more better molecules in hand, we wanted to ask, are they right for the right reasons? And so Chenghua Wu, a postdoc in Yorgo's lab, determined the structures by cryo-electron microscopy. What I'm showing you, remember there's 2 monomers, so we have to predict them in both. This is the active monomer here, and this is the inactive monomer. And the blue guys is the docking prediction, and the pink guy is the cryo-EM result. And so basically here too, were the docking prediction superimposed with the experimental results. There were some differences. You can see that this ring swings down. And interestingly, and I'll come back to this in a second, there were differences in the structure of the receptor. We hold the receptor rigid in docking, but in reality, it can move. And sure enough, there were some important conformational changes in both the positions of some of the helices and rotamer changes of individual amino acids. This molecule, the second molecule, is the one we're going to take into animals. And here there were actually slightly bigger changes. The molecules are going in the right place. They're both going into the allosteric site as predicted. There's pretty good correspondence in this monomer A. The monomer B, still most things are in the right place. The cation's in the right place, interacting with the right residue. This sort of anchoring group here, this group here, is in the right place. But this thiazole, this benzothiazole ring, has swung in the experimental structure relative to the docking post. So there, the fit is a little bit less perfect. And once again, we're seeing these pretty important rotations of individual amino acids. The other thing we see is that the overall structure of the receptor has changed a little bit. I'm showing you a superposition of the complex of the G protein, of the like key residues and key helices in the receptor, in the cinacalcet bound mode in this sort of light whitish color, and then compared to our new molecule in orange. And this key transmembrane helix 6 has changed its position, and it's adopting a position that's more akin to what you see in the fully activated state of the receptor. So it seems that So these structures were determined without G protein present. And when G protein is present, the receptor is fully activated. And I guess what I'm trying to say here is that in our structures, the receptor, even though the G protein isn't present, has adopted a more activated like state. And we thought that might be important. So we took the new molecule, this one molecule, 54149, into in vivo like studies, and then eventually fully in vivo. So this is an experiment on isolated parathyroid glands, well, parathyroid glands that are, ex vivo assays on the gland. What you can see, what's been shown here is the concentration of the drugs, cinacalcet, evocalcet, and this new molecule. So cinacalcet just look at cinacalcet for a second. It's in green, and you can see that it's actually relatively weak in this assay. And this new molecule is actually, in terms of EC50, a lot stronger and leads to much higher suppressions of parathyroid. What you're trying to do is suppress parathyroid secretion. And you can do this assay a different way. This is holding calcium constant and changing the concentration of the allosteric modulator. But you can do the reverse. You hold the drug constant, change the calcium levels, and you get the same results. So the white is vehicle here. This is and increased calcium. So here's no allosteric modulator. This is with when cinacalcet is the allosteric modulator. This is evocalcet in gray, and then our guy is in orange. And so you can see it's left shifted, and it starts already, at the earliest, the lowest calcium level, which is around half a millimolar. You've already got meaningful suppression of parathyroid hormone secretion because the molecule is potent. So then we went fully in vivo. These experiments are all done by Wenhan Chan at UCSF. So first we established that the molecule was exposed in pharmacokinetic assays. So this is the blood PK, the plasma PK, cinacalcet at 3 mg per kg. This is our molecule at the same dose, so it reaches higher exposure levels. Evocalcet is actually, the pharmacokinetics are much better, at least judged by exposure. So you can see that. But anyways, so then we took both cinacalcet and this new molecule, this docking-derived molecule, 54149, into in vivo studies. And what you can see is that at the highest dose of cinacalcet and the new molecules, both of them really fully suppress parathyroid hormone secretion into the plasma. But if you go down just half a log, you can already see that for cinacalcet, the plasma levels of parathyroid hormone are significantly higher. And meanwhile, the new molecule, they're still completely suppressed. And that's true also at 1 mg per kilogram doses. And so cinacalcet rises again. Our guy starts to rise at 300, that's 0.3 mg per kg, but it's always meaningfully lower, significantly lower than cinacalcet. So in a dose per dose range. So this is also consistent with this new molecule being more potent than the established drugs. And then perhaps the most interesting, and I have to admit, something we can't fully explain at a molecular level, but it's really something we were looking for, is that the new molecule does not lead, at least in the mouse studies, to severe reduction of calcium. And that's the problem with cinacalcet and evocalcet, is you can see it here. This is dosing at 3 mg per kg cinacalcet, and then watching the calcium levels over time, and you can see they drop. And this is what leads to the toxicities of this drug. But with the new molecule, they basically stay unchanged. They stay within the physiological range. We don't actually fully understand why, but it's great. That's definitely the one thing we can say is we were looking for new chemistry, hoping the new chemistry would confer new pharmacology, and that turned out to be true. I notice I'm running low on time, so I'm not going to finish the full story. I'll tell you about one of the problems we've encountered, and what I hope it's very new work, certainly unpublished, but I think it speaks to the challenges of ligand optimization in in drug discovery. So going back to this sort of trophy chest that I showed you at the beginning, what you see in every one of these cases where we're optimizing these molecules and taking them into animal studies, that 2 arrows. What 2 arrows means is what we got the docking hits, those are the first arrow, and then a lot of work went into optimizing the molecules for efficacy in animals. And when we looked back historically on, these campaigns and asked, well, how good are we at making an analog and getting a big improvement in activity? It turns out we're not very good at it. So we set like a meaningful improvement is tenfold. Make an analog, any analog that you want, and how many of them improved affinity by tenfold or better. And in all of these cases, we were trying to improve affinity. And it turns out that like in less than 5% of the cases was a single analog tenfold or better than the parent compound. So, I think not a great success rate because, we're using structure-based design and sometimes free energy perturbation and all of our tricks. And still, less than 5% of the time do we get a big jump, a big improvement. It's often much more incremental. And so one of the things we started to wonder was, well, is 5% good or bad? And we didn't know, and nobody knew. Like if I would go, or I still do this, I go around and ask medicinal chemists, what percentage of the time would you expect to get a tenfold improvement? And actually, usually the numbers are like 1% of the time or 2% of the time, but nobody knows that. Like, there's no background set for medicinal chemistry. There's just opinions. And so we didn't know if 5% was good or bad or indifferent because we've got nothing to compare to. So we thought, okay, why don't we try and develop a model for what we should expect at random? Because in your bioinformaticians, that in bioinformatics, all comparisons are to background models. So that's how BLAST works, right? You have a background model for expectation of sequence identity, for instance. And that's where the evals come. There's nothing like that in medicinal chemistry. So we thought, well, maybe let's try and make one. So this is what we did. We thought we would, starting with 18 scaffolds, each scaffold is a different core structure for a starting ligand. Active, I say 5 targets, actually 6 targets. Let's make 350 small changes. So we're going to make 350 analogs starting up with 18 different starting points, active on 6 different targets. And the differences are going to be very small. So we'll start with, like, for instance, this molecule, and we'll convert like a carbon one at a time to a nitrogen. And we'll just do that systematically and comprehensively around the ring for all the analogs that we can make. Like there's some molecules that are too expensive, so we wouldn't make those. And then we would take the hydrogens off this ring and convert them into methyls or fluoros or chloros or sometimes hydroxyls. And so each analog has only a 1 atom change, and it's a small change. And the question was, well, so with this, we say, okay, this is a systematic, more or less comprehensive set across a diverse number of targets and a diverse number of starting points. How often do we see tenfold or better improvement across this set? And we don't have the full results yet. but I'll show you where we are. And this is just to give you an example. Here's this molecule now, and these are all the changes we made in this molecule that we were able to make. So like, for instance, at this position, we put in a chloro, a hydroxyl, and a methyl here, one at a time. And none of them were better than 10-fold. But over here, we got closer. Like, oh no, so I should go over here. Here we put a methyl in and it improves things 18-fold, for instance. Okay, so that's an example. And these are the data that support that. So full dose response curves. Okay, so this is a summary of what we've seen so far. So this is against the alpha-2A G protein coupled receptor. And these are the number of analogs that have been made and tested. This is against the cannabinoid receptor, the mu opioid receptor, so all GPCRs, serotonin transporter, so now a completely different family. And this is against beta-lactamase and MAC1, so these are two soluble enzymes. So so far we've made 278 analogs, and depending how you calculate this, about 11% improve by 10-fold or more with these small changes. So in this sense, this random sort of background set is better than we are when we're trying. And I think a lot better than most medicinal chemists would expect. So we would say this is starting to establish a background for what you can expect in optimization, at least with small changes. The other thing I'll say is that the landscape is frustrated, however. So if you think of protein folding funnels, this is an analogy to that. Protein folding funnels are not frustrated. That's why proteins fold. But here, if you say, well, for medicinal chemistry, you don't just want to improve affinity, you often want to improve fraction unbound, stability in the plasma, stability to liver microsomes, whether they are permeable, and whether they inhibit the hERG ion channel. And so we did those experiments too. Well, we paid a CRO to do them. And this is just one example, and I'll show you this in more detail in the next slide. But what we found is, whereas like we could get molecules that had 10-fold or 20-fold or sometimes better improvements in activity, it was rare for that molecule that was better to also be better in these other terms. Sometimes it was a draw, but sometimes they were much worse. And that's something. So this is just one molecule. This is over all the molecules where we got improved activity, and it was really rare for any one of these 10-fold or better active molecules to also be improved. Often they were worse. Typically they were worse in these other crucial criteria for whether a molecule is going to be biologically active or not. And it's the same story for, if we say, okay, well, what about 3-fold improved activity? I think, well, one of the conclusions here is that actually you can have an expectation for how well you should do in medicinal chemistry. And I think the expectation is at least 2 times better than we are when we're trying, just at random. But then the other side of the story is that, improving a molecule from being active on target to being useful as a therapeutic or an in vivo active probe is very difficult, because not only are these different criteria orthogonal from affinity, they're often opposed. So I know I'm being slower in this talk than I meant to be, and I do have I have to go soon. So maybe I think I'll just leave you with that thought, and the conclusion is you can get as the libraries grow, we're getting better and better results, and so one of our conclusions is just make them bigger. That's a big focus of our lab, and the new chemistry that you get out of the large libraries can confer new biology, which has always been the hope of structure-based discovery. So we're super excited about that. And then the challenge is that drug discovery remains a challenging area. There's huge opportunities in it, but optimizing a molecule for all the properties that it needs, we still need new insights. And so that's why we need young, brilliant people like you to come into the field. So thank you very much.
Thank you, Brian, for the great talk. You have like 3 minutes, right?
3 minutes,
Okay. So there is a question here in the chat from Danielle Guzmão, which is, how can the field of statistical mechanics inform the study of molecular docking?
I think StatMech can be, well, we use it at a primitive level, and yeah, it could be hugely helpful. For instance, sampling new states, important states, you saw that we undersampled them for the receptor and weighting them according to, a Boltzmann distribution. That's where the StatMech would come in. That would be super helpful. It's tricky right now. Because we're not sure we're sampling the right states, and sampling the wrong states can be a big problem for docking. But for sure, it could be useful, and there's a lot of interest in doing that.
Okay. Also from Alex, do you believe that AI can improve docking results?
I think AI, like what we've convinced ourselves is that bigger is better. I didn't get to that part of the talk, but we haven't the paper that just came out last week from Feng Yu on CASR speaks to that, and we have some new results that I didn't show you. So we're quite convinced that as the libraries grow bigger, we get better results. The problem is that we can do 6 billion brute force, which is what we're doing now, but now we want to do like 6 trillion. And that we can't do without a guided search through the library. And that's, I think, one of the places that AI, pattern matching can really help. Whether it can improve scoring functions, which is where most people are focusing, actually, I'm less confident about, because it's a really intractable problem. But I think clearly in the area of just getting to larger spaces, we have to go to machine learning. And there's a lot of work going on there in our lab and in other labs.
Thank you. One question from Vinicius Bonato. Do you believe that ultra-large docking is the most effective approach for discovering new chemotypes, scaffolds in current drug discovery efforts?
Is the most effective method for doing what, Rafaela?
For discovering new chemotypes, scaffolds in current drug discovery.
Yeah, I mean, I don't like to say it's the best method, but it's a very effective method. And there's other methods for getting, really novel chemical matter that use a large library approach, like DNA encoded libraries. Very exciting. But I think that molecular docking these days, the ultra large library docking, is among the best ways. I don't like to say it's the very best, because there's all these wonderful new technologies.
Well, thank you so much for being here with us today, Brian. I don't know if Miguel or Tetsu want to make any final comments.
Yes, Brian, I know that you have to go, so I want to just a little comments. We had an audience size of approximately 80 participants here. So I think that your talk had
also some 30 people in YouTube, so over
over 100. So I think you had inspired many people here in Brazil. So I want to thank you for your great talk here in our event. Miguel, do you want to say something?
Yes, just thanks Brian for this wonderful Wednesday for us, and it was inspiring, and it's going to be a pleasure to have you in person someday here in Brazil for a meeting. Let's plan to do that. Thank you.
Thanks, guys. I really enjoyed it. Next time in person.
Great. Thank you. Bye.
Bye, bye.
Eu tinha umas 30 perguntas, mas tudo bem.
Eu também.
Ah, pois
Mas eu
é,
privilegiei as que não eram minhas.
mas foi ótimo, foi ótimo.
Bacana.
Ô, Miguel, qual vai ser o próximo?
Então, eu não coloquei na página, mas mês que vem, eu acho, é de sexta-feira, e a gente vai ter o coordenador da linha Manacá, o coordenador do Sirius, simplesmente o cara do Sirius falando para vocês aqui.
Eita, legal, legal.
Joia.
Então fica o convite aí pro pessoal. Então é no mês que vem, Miguel? Na sexta do mês que vem, no final do mês?
É praticamente daqui a um mês, que tá um por mês, assim. Mas se a gente achar mais algum, a gente coloca aí também, né? Mas era para eu ter colocado no site hoje cedo, mas não deu, não. Eu estava indo comprar meu carro aqui, ó, Tetsu, porque o meu up eu sair da estrada e bater no guarda-reio, porque né,
Ah,
falou 3 vezes e eu tentei segurar.
que
O outro,
isso?
agora vou baixar uma playlist de rock, porque sexta-feira eu vou estar andando por aqui de Rock in Rio vermelho.
Tá certo.
A carona aqui vai ser de Rock in Rio, Tetsu. Pode trazer os tampões de orelha aqui, porque a música está alta.
Tá certo, Miguel. Entendido. Bom evento lá para você.
Valeu, gente. Todo mundo esteve hoje em Natal, né? Organização prioritária aí da UFRN. Não vamos sair para comer camarão com o Tetsu, porque não come camarão, mas quem sabe a gente vai procurar um peixinho aqui para comer no almoço agora. Né?
Ah, mas acompanha com outras coisas.
Fim de companheiro de sushi e sashimi, né? Aí sim.
Isso, pronto. Tá certo, então. Obrigado.
Não é só Yanará que fala. Como é que é?
Sayonara mesmo. Tchau.
Tchau. Valeu, gente. Como dizem os... Como chama? Os Teletubbies. Hora de dar tchau.
Tá certo. Até mais então, Miguel. Até mais, gente. Até a próxima, então.